Medicine Without Cruelty presents Understanding Human Biology. And this is part one in the series because understanding human biology is the key to non animal science. To recap the last video, animal testing is outdated, ineffective, and ethically, we cannot defend it as a practice because it has no place within science. When you have alternatives like organochip, AI driven models, 3D bioprinting, and organoids, to name but a few, which we'll go through in subsequent videos, we have the tools to conduct research in ways that are accurate, reliable, and compassionate. Educational institutions hold the key to driving this change because it will be our students who will embed these practices, and human relevant models will become commonplace, as will treatments for human health. Okay, so all humans are made up of cells, tissues, and organs. Yes, all animals have cells, tissues, and organs, but the complexity of these structures can vary so significantly across different species that fundamentally animals don't replicate human biology, and that's a massive problem. And I hope to explain that in subsequent slides. So let's get into cells. Now, cells are the smallest biological unit and they make up all living beings. I've got this picture of Lego bricks there on the side, and I want you to think of cells as these tiny Lego bricks. They might vary in shape or color, but when they come together, they can make different structures that have different functions. Now, cells can communicate with one another. They can tell each other whether a substance is okay to absorb or not. They can signal different things to one another. So, cells are the fundamental building blocks for all living organisms, and that's whether it's plant or animal. And I'm just going to go into what animal cells look like, specifically human. Now, all cells have a variety of different components, and these components are known as organelles. These organelles can be mitochondria, the nucleus, the cell membrane. We have tons of more organelles within the cell, but just for this mini video, we're just going to cover the three most important ones. Now, the cell membrane, or that kind of outer wall of the cell, that's known as the gatekeeper. Now, it's not as rigid as a wall. I want you to think of it as kind of a wall with many doors. Now, this gatekeeper controls what enters and what exits the cell, and it plays a very important role in drug absorption, distribution, and cell signaling. Like I said, cells communicate to one another. So cell signaling is a very important process because it can begin different processes, produce different functions within the cell. And it's really important because non-animal methods like tissue engineering or 3D bioprint use human cells to study how drugs interact with the cell membrane, especially how they're absorbed and how they affect cellular communication. Because you want to be able to create a drug, we're talking about medical drugs here, that is safe for human beings. And so we want it to be able to target the right part of us. So whether they are absorbed and how they, whether they positively or negatively affect cellular communication are critical factors to consider for drug development. And remember, human cell membranes react very different from those of different species. And so if we model this in human relevant systems, scientists can obtain accurate data on how effective a drug is and also any potential side effects. The cell nucleus, this big structure there in the middle of the cell, is known as the control center, and its function is simple. It contains the cell's genetic material, the DNA. And you might have heard of DNA before. The genetic material refers to these special molecules that carry all the information needed to control the activity within a cell and within a living being. This information dictates everything from physical traits such as hair color, skin tone, eye color, or even facial features, for example, to how the body functions at a cellular level. So our genetic material codes for everything. And they're packaged within these what's known as genes. And these genes will be expressed, and that just means that it will express the particular physical trait, for example. So if we take the example of hair color, that they will be parts of your genetic material that will be packaged within a gene that tells your body produce brown hair, for example. So it's very important, the cell nucleus, for regulating this, and also cell division, because our cells do divide and proliferate. It's important to non animal methods because human relevant models, such as human cell cultures, and just to remind you what a cell culture is, cell culture refers to the removal of cells and their growth within an artificial environment. So we're taking cells, so say if it might be a blood sample, it might be even if we swab our cheek, we can obtain cells from that method. Those cells can then be put in an artificial environment that's good for it. So remember, our human body operates very well at 37 degrees. So we need to make sure that we put these cells in a nice warm environment, and we provide it with the nutrients, the relevant factors needed to help it grow. These human cell cultures can be used to observe how treatments or environmental factors influence that gene expression directly within human cells. And it's really important because organoids and organoid chip systems can model how gene expression in the nucleus might affect a organ's function. And it means that these methods are predictive of human health outcomes. Now, finally, for this part, we have the mitochondria. Now, our mitochondria is the powerhouse of the cell. The function of it is simple. It generates the energy necessary for cells to function. And it plays a very key role in human metabolism and therefore drug metabolism as well. Now, the biological, metabolic, and even genetic differences between animals and humans make animal models completely unreliable for detecting if there's any sort of drug toxicity within the mitochondria. And that's why it's really important that we have human relevant models such as cell cultures or organoids to study these effects. These allow researchers to observe how drugs affect mitochondria, providing accurate data. Now, sometimes, animal models often miss, mitochondrial damage due to potential drugs because of the metabolic differences between these species. And yet another reason why we need to make sure that we have human relevant models.